Hi, I'm Anand Ramani. I basically work at Innocent Meat GmbH, which is a cultivated meat industry. My main role is basically as a research team lead in the expansion media development, where I try to focus on isolation of cells, characterizing them, and develop serum-free media for the expansion of these cells. In cultivated meat, we need to get cells from the animal, we need to expand them, and then differentiate them either to muscle or fat. So, my department, I'm basically responsible to grow these cells in mass in order to differentiate them towards fat or muscles. My journey started at Innocent Meat in 2021. Back then, I was basically an one factor at a time analysis kind of guy, where I tried to analyze the role of each ingredient in the media, but one factor at a time. It's basically a long process. You just change one ingredient and you try to test what is the implication of this change on the growth of the cells. So that is when we were introduced to DOE, design of experiments, and we came across Synthace. Basically, I had no clue about this concept of DOE. And back then in 2021, this was also not very well established. So this is why when we got to know about this concept, we thought that it is very innovative, it is very powerful, because when you talk about media development, there are many ingredients to test. And just testing one factor at a time is a long process, and you waste a lot of time and resources. So that is why we wanted to venture out into learning DOE and what is Synthace able to provide in the form of DOE. And now it is something that we design on a platform that Synthace provides. And that is something very intriguing for us. So we can plan really complex experiments on Synthace. Not only plan, but also execute it. Just recently, we have executed an experiment, and it feels like we would be able to learn a lot from these media changes and see what happens in terms of reducing costs, in terms of effective growth, as well as effective duplication rates. So basically, when we speak about growth rates, this is something that concerns the industry. You want cells to grow fast and with less budget. So I think Synthace is capable of achieving both these parameters.